your other screen.
Sure. Okay. Welcome to everyone, and thanks for joining our Liver Cancer Program Seminar Series today. We're happy to welcome Dr. Lewis Roberts from the Mayo Clinic, and we're looking forward to his presentation. Shin will formally introduce Dr. Roberts, but just a quick note that we have an upcoming Liver Cancer Program Seminar this coming Monday on June 7th. It'll be at 9 o'clock in the morning Eastern Standard Time. And Shalav Itzkovich will be our speaker from the Weizmann Institute. He'll be speaking about spatial omics of mammalian liver. And for today's seminar, if you have any questions for Dr. Roberts, you can add them in the chat box, or you can turn your mic on at the end of the seminar and ask your question then. So that's it.
Wonderful. Thank you, Anu. As I want to extend the same welcome to everyone for this NCI Liver Cancer Program Wednesday Seminar Series. My name is Xinwei Wang. I'm the co-director of this program. Now, on behalf of a colleague at the NCI, it is obviously my distinct honor to introduce today's speakers, Dr. Lewis Roberts from Mayo Clinic. Now, a few words about Lewis, who is a physician scientist and is a Peter Francis George Son Professor in Gastroenterology and Cancer Research and consultant in gastroenterology and hepatology at the Mayo Clinic, where he's also co-PI of the Mayo Clinic Liver Cancer School, among many, many other academic and administrative titles. Lewis earned his medical degrees from University of Ghana Medical School, a PhD from University of Iowa, and further training in internal medicine, gastroenterology, hepatology, and cancer genetics at the Mayo Clinic. Now, Lewis' practice is focusing on liver and biliary cancer and the GI endoscopy. His research focuses on molecular mechanism of liver carcinogenesis biomarker for diagnosis of liver cancers and for prevention, diagnosis, and treatment of hepatitis liver cancer. He has been consistently funded by NIH, as many, many other funding agencies and foundations, and has been published over 400 publications, including book, chapter, and also edited a book called Evaluation and Management of Liver Cancer. Now, Lewis has a very strong interest in cancer health disparity and is committed to reducing the burden of illness and death from liver cancer in the United States and as well as globally. He also has a strong interest in mentoring fellow in science, having mentored over 190 student fellow with a very diverse background over past 23 years. And many of them now at faculty at Stanford and Yale and the UCLA, Cedars-Sinai and Mayo Clinic, etc. Now, currently Lewis is deputy editor for hepatology and the editor board of liver cancer. He has received many, many awards for his accomplishment, which I'm not going to mention today, except a few noticeable. A Distinguished Alumni Award for Achievement from the University of Iowa, Teacher of the Year for Mayo Clinic Graduate School of Biomedical Science, Fellow of the African Academy of Sciences. He also served as the president of Africa Partner Medical, a non-for-profit organization focused on improved healthcare delivery in Africa through medical education, practical skill training, and provision of medical equipment and supply and health advocacy. So without further ado, let's all welcome Dr. Roberts.
Thanks very much. And I'd just like to thank Dr. Budu and Dr. Wang for the invitation to present. So I thought I'd talk about mostly about cancer biomarkers for HCC. And at the end, I'll also discuss applications or what cancer biomarkers might tell us about genomics of cancer in a recent paper that we published with one of our colleagues. I think that's just been accepted. I have a few disclosures. So I thought I'd start with a couple of patients to just sort of set the context. So this first patient, I think I saw him maybe about 1.5 years ago, was a 60-year-old male. He had immigrated to Minnesota from Vietnam at the age of 30. And he remembered that he was told that he had hepatitis B when he first immigrated to the United States, but no treatment or surveillance was recommended at the time. When we did test him, we found that he was hepatitis B surface antigen positive, so he had chronic hepatitis B, but he was E antigen negative and E antibody positive, which sort of defines a person who typically does not have active ongoing replication of the virus. His HBV DNA was 160, so it was relatively low. But in further questioning, it turned out that he had additional risk factors as well. So he was a 40-pack-a-year smoker, and he would drink 6 to 8 beers per day. He had presented with a 1-month history of abdominal pain, and when he was evaluated locally, he had a CT scan that showed a large mass in his liver. And then we get into biomarkers, because when we assessed him for a number of biomarkers, we found that his alpha-fetoprotein, which is the most common biomarker we use, was normal at 3.5. But the AFP-L3, which is a lectin-binding isoform of AFP, which is typically more specific for hepatocellular carcinoma, was 23, which is elevated. And his des-gamma carboxyprothrombin, which is another biomarker, which is a modified prothrombin, typically more specific for liver cancer and particularly for more advanced forms of liver cancer, was fairly highly elevated at 425. We'll talk about these biomarkers later. So when we did the imaging, you can see here the fairly large tumor in his liver here, in the right part of his liver. This is his heart, so this is sort of a cross-section through his body coronally. And here's a different patient where fever, weight loss, and fatigue were what triggered imaging. There's a 41-year-old male, and he had also recently immigrated to Minnesota just a couple of years ago, but he had immigrated from West Africa, and you note the difference in age of these two patients. He presented locally with fever, weight loss, and fatigue. In fact, since he had just recently come from Africa, the initial thought was that perhaps he had malaria. But further investigation showed that, again, he had hepatitis B. He was hepatitis B surface antigen positive, so he had chronic hepatitis B. He also was E antigen negative and E antibody positive. And he actually had only a detectable HBV DNA. So very low, you could tell he had HBV DNA, but was very low. Again, a CT showed a large mass in the liver. In his case, he had an elevated AFP and AFP-L3, but the DCP was normal. And there's a large tumor in his liver that shows the extent of cancer in his liver. So worldwide, there is estimated about 300 million people with chronic hepatitis B, and chronic hepatitis B worldwide is the major cause of liver cancer. And this shows the numbers in different regions of the world. We can see the Western Pacific and Southeast Asia have fairly large proportions of the global burden, about half the global burden. Africa has about a fifth of the global burden, and then we have other countries and regions with different additional proportions. If we look at the global epidemiology of liver cancer, we see that to some degree it mirrors the epidemiology of HCC. Or the epidemiology of HCC mirrors the epidemiology of hepatitis B that I just showed you, with high prevalences or high incidences in Asia as well as in Africa, and then intermediate incidences you can see in Europe and North America. You have to point out Egypt, which is the country worldwide that has the highest prevalence of chronic hepatitis C. And so Egypt sort of stands out because even though hepatitis C prevalence generally across the globe is lower, Egypt has high prevalence and a high rate of liver cancer from this. In the United States, liver cancer is the major cancer that is actually rising in incidence, so that for most other major cancers in the United States, we actually are seeing declines in incidence. And you can see here the estimates from 2020s here out to 2040. We actually expect substantial reductions in lung cancer. Pancreas cancer is about stable. And then colorectal here and breast cancer are declining. But we see this increasing trend for liver cancer, and the estimate is that liver cancer will overtake colorectal cancer actually somewhere around 2035 and become the #3 cause of death from cancer in the US. And if we look at different communities within the United States, what we see is that Asian and Pacific Islanders, people of Asian Pacific Island race, have the highest, or had, I should say, had the highest incidences for many years. You can see the decline here, and this goes up to 2012 and it's continued to decline. Hispanic Americans have had a rising incidence. The same is true of African Americans and of Caucasian white Americans. Of course, whites actually have the largest population in the United States. So overall, the largest number of cases of liver cancer in the United States are among white Americans. If we look regionally at the United States, what we see is that there's a fairly substantial regional variation. And we can see here that regions of high population density, but also regions that have higher proportions of Asians and Pacific Islanders, of Hispanics, and then also of combination really in the Northeast of Asian Pacific Islanders and people of African descent tend to have higher rates of liver cancer. But what we see also is that even within states that overall have relatively lower rates, we have often pockets of high incidence of liver cancer. And we see one, for example, I don't think I have a slide of it, but typically, for example, in Minnesota, we have fairly large populations of immigrant Asians, as well as immigrant Africans in larger cities and intermediate-sized towns. And so if you looked at the Minnesota map, for example, a detailed map, you'd find that around Minneapolis, around Olmsted County, Minnesota, where I live, we have actually fairly high rates of liver cancer. In fact, we're just in the midst of finishing an update of liver cancer rates in Olmsted County, in this county, and we actually have rates that are around 15 to 20 per 100,000 per year, which would make us a high incidence country if our county was a country by itself. So the question about biomarkers really relates in large part to the potential benefits of biomarkers in hepatocellular carcinoma surveillance, and the idea that we can identify people who are at risk for liver cancer. And if biomarkers allow us to survey them for development of liver cancer and potentially identify liver cancers at an earlier stage where we can treat them more effectively. And even though there are relatively few randomized control trials that show the benefits of surveillance in patients at high risk for liver cancer, there's a substantial amount of global real-life evidence that supports the benefits of surveillance. And this study that I'm showing here, the BRIDGE study, is one such study where we looked at global patterns of hepatocellular carcinoma management from diagnosis to death a number of years ago. And so the BRIDGE study involved 42 study sites on 3 continents, primarily North America, Europe, and Asia. And what we saw was that if we looked at these different regions and countries, there was a fair bit of variation in the risk factors for liver cancer. So you can see, for example, here in North America at the time, Hepatitis C virus was the predominant cause of liver cancer. We had about equal proportions of hepatitis B and alcohol, and we actually had much lower rates of fatty liver disease-associated liver cancer. Today, fatty liver disease-associated liver cancer really is about equal to hepatitis C virus, and we have seen some declines in hepatitis B virus-associated liver cancer. You can see in Europe, it's mostly hepatitis C and alcohol. In China, mostly hepatitis B, the combination of hepatitis B and hepatitis C in Taiwan. In Korea, mostly hepatitis B, and then in Japan, mostly hepatitis C. So when we looked at the numbers of patients, and what we did was we looked at patients that were being seen at major medical centers in these regions. And we weighted this in favor of Asia because so much of the global burden is in Asia. And you can see the different countries and regions and the different proportions of cases. We had a total of 18,000 participants that were enrolled in this study. So what we found when we looked at the stage of diagnosis of these patients was an interesting observation, which is that globally, most HCCs are diagnosed at more advanced stages, at either intermediate or advanced stages, except in Taiwan or Japan. So here you can see the BCLC stage where we separate this as very early, intermediate, advanced, or terminal stage disease. And what you'll notice is that in North America, Europe, China, South Korea, we found 63 to 70% of cases were being diagnosed at intermediate, advanced, or terminal stage disease. On the other hand, in Taiwan and Japan, we had 70 and 73% of cases diagnosed at very early stages. And then we asked the question, what were the survivals of these patients who had been diagnosed at these centers? And what we see here is that in those two countries as regions, Japan and Taiwan, where we had relatively early age, very early stage of diagnosis, we actually had exceptionally good survival. So if you look at 5-year survival, 5-year survival was actually around 70% in Taiwan, and 5-year survival was about 50% in Japan. In most of the rest of the world, what we saw was here in the United States, for example, North America, about 33 months for median survival. And then in Korea and Europe, we had 23 months for median survival. Now, this was not part of the study, and I'm not going to show you the data, but we've performed a number of studies subsequently in Africa with colleagues there looking at median survival. And in sub-Saharan Africa, there's almost no surveillance. In the one study we did, only 3% of individuals diagnosed with HCC were diagnosed when they were under surveillance for cancer. And consequently, pretty much everyone is diagnosed with advanced disease, and the median survivals are 2.5 months. So this is what I call the surveillance gap. Between Taiwan, Japan, and Africa, you can see the difference in outcomes. And a major part of this is the fact that in both Taiwan and Japan, every adult gets screened for chronic viral hepatitis. Those individuals who are found to have chronic viral hepatitis and are at risk for liver cancer are placed in a national surveillance program, and they'll get surveillance anywhere from every 3 to 4 months in Japan for people considered at highest risk, to every 6 months in Taiwan, which is more the typical global standard. And they get this surveillance on a regular basis. And you can see the difference that surveillance makes. So it's important then to define the need. The need in many parts of the world is for advances in surveillance and early diagnosis in order to improve outcomes of patients with liver cancer. And we then, of course, need advances in treatment as well, to improve the outcomes of those who are diagnosed with intermediate to advanced stage, or those who are diagnosed early but have biologically aggressive disease. Then in addition, we need advances in treatment selection. We know that for almost every treatment we've identified, not all patients will respond equally. And so it's important that we are able to identify the pathomolecular features that allow us to optimize the outcomes for individual patients while minimizing toxicity. So what I'd like to talk about now is some general concepts of cancer biomarkers, talk a little bit about different utilities of cancer biomarkers, and then a validation strategy that's been developed for assessing different biomarker assays, talk about some of the challenges of the use of biomarkers in clinical practice, and then discuss some of the current and future HCC biomarkers, identifying at-risk populations, surveillance, early detection and diagnosis, and then prediction of treatment response, and a little bit about prognosis as well. And we'll talk maybe about some future directions. And if there's time, I'll actually talk about a future direction that is more that is applicable more to cholangiocarcinoma, but I think tells us where maybe we should be headed in terms of HCC. So general concepts of cancer biomarkers. I'm not sure if this will work. And I think I'll just skip it. And I think the key point here is that there are different types of molecules that are in the bloodstream that we can use as biomarkers. It's possible to use DNA and look for mutations and other alterations in DNA, such as methylation, which we'll talk about. It's possible to look at for RNA molecules or microRNA molecules. These molecules might be organized into vesicles, for example, and people that are looking at vesicles as a diagnostic platform. There may be proteins that we measure, such as alpha-fetoprotein. Many of these proteins might be post-translationally modified, for example, and it's also possible to use metabolites as biomarkers. So utilities of biomarkers. So we've talked a little bit maybe about the idea that we can use biomarkers to identify the population at risk. So diagnostic biomarkers, for example, for viral hepatitis, fulfill this need, but it's possible to identify other biomarkers as well. In actual fact, the three biomarkers I showed you, the AFP, AFPL3, and DCP, are actually approved by the FDA. The AFPL3 and DCP particularly as risk biomarkers, not necessarily as diagnostic biomarkers. But we can use biomarkers for surveillance, for early detection, for figuring out who has cancer out of the sea of people at risk who do not have cancer. And often, if we can use biomarkers depending on the level for diagnosis as well. And often, if you use a high enough stringency at the level of a biomarker, you can use it as a diagnostic tool. And then we can use biomarkers to predict treatment response as well. So these patients with cancer, which ones are most likely to respond to treatment? And we can hopefully use them to also to determine who's likely to do well versus those who might not do well on treatment. So I'll talk next about the validation strategy for biomarker assays. And this was really first, I think, most clearly articulated by Margaret Pepe and her colleagues. And it really breaks down the phases of cancer biomarker development into 5 phases. And I'll just go through these different phases. So phase 1 is sort of the preclinical exploratory phase, where we have a tumor in the adjacent benign tissue. And we compare genes, proteins, metabolites, or perhaps cancer-specific antibodies between case and control tissues. The next is the clinical assay development and validation phase. And so this is where we take tissue or serum or urine or plasma, and we first of all develop an assay that's based on this preclinical exploration and say, do we measure a particular protein or metabolite at higher levels in patients with cancer versus those without. We ended up wanting to validate them in the laboratory and then between different laboratories to see if these assays can be replicated rigorously. The next phase, which is often done, is the phase where we do what we call a retrospective longitudinal study. So this involves prospectively collecting samples from patients in the course of their care, collecting the samples in individuals at risk and through their diagnosis of liver cancer, and then coming back and examining those samples to determine whether our assay would have been able to detect the cancer, in this case liver cancer, before the cancer was clinically detected. And so it gives us an idea of how well would this have performed if it had been part of the management algorithm for the patient. Phase 4 then is a prospective screening study. It is the idea that we then randomize individuals to be screened with a biomarker versus not screened with a biomarker, and then ask the question, do we detect cancer more frequently or at an earlier stage in those who are undergoing screening versus those who are not undergoing screening? And then finally, phase 5 is really a cancer control study. It asks the question, if you apply the biomarker in a population under screening, you get better long-term results and less mortality in the population compared to the population that did not have the biomarker performed in screening. In practice, we see a lot of phase 1 studies. We do for the potential biomarkers of interest phase 2 studies. Unfortunately, that's where a lot of studies end, is at phase 2. It's much more challenging and takes a longer time to do phase 3 studies that are of course more rigorous and we learn more. And phase 4 and phase 5 studies are really, really difficult to do, and we've seen very few studies in the liver cancer space, for example. So I'll talk now a little bit about challenges of biomarkers in clinical practice and the use of biomarkers. The first one is that people are heterogeneous. People are different. And so it is difficult to establish a negative baseline value for any one biomarker because people tend to have different levels of any one biomarker. The second issue is that cancers are heterogeneous, beyond the fact that people individually have their differences. And cancers, these are all liver cancers, but they all look very different. And because of their differences, they'll express different proteins, different messenger RNAs, different metabolites at different levels. So the ideal biomarker would be able to separate, people who are at risk or controls perfectly from cases. And if we do this area under the ROC curve, we would get, comparing specificity to 1 minus specificity, we get an area under the curve that would be almost 1. In practice, there's overlap. So most real biomarkers show overlap between the cases and controls. So you can see here are controls that have high levels, and here are cases that have, levels that overlap with those of controls. And that means that it's difficult to separate cases from controls. What's the ideal solution? It would be thinking from the perspective of having each individual be their own control. So we deal with the issue of individual variability and the ability to use all possible biomarkers. So that if we had all possible biomarkers, then for any one individual, we could potentially find a biomarker that for them would be elevated when they had disease. And then hopefully we would treat the disease and we would see a drop in that biomarker. So that's the dream. And there's actually people that are working on this as a dream. But what's the feasible alternative? What is it that we can do now? What we do now is we take combinations of the laboratory tests, clinical risk factors, and we look for different types of reagents. We look at the alpha-fetoprotein, we look at metabolites, different types of RNAs, DNA copy number changes, or mutations, or methylation. And we try to build a diagnostic model that works better than any one of the individual components. So I'll talk now about some of these models and models that we've used to identify at-risk populations, things that we're doing to try to improve surveillance for particularly HCC. And then we'll talk about a little bit about predicting treatment response. So here's a model for predicting HCC risk in HBV in infected patients. And so this was developed and includes viral factors. So in the HBV DNA, the surface antigen level, the HBe antigen status, the HBV genotype. It contains patient factors, the patient's age, gender, whether or not they have a family history of HCC. And a laboratory value, the alanine transaminase, which is an index of inflammation in the liver. And you can see here that this allows you to build a model where, depending on the sum of the risk score, you can actually determine what a person's 5-year risk of cancer is. So if this person had a risk score of 2, you can tell that this is their 5-year risk of cancer, their 10-year risk, their 15-year risk of cancer. And you can see that across this range of risk scores, you can actually have substantial, you can have orders of magnitude of differences in HCC risk. And so this can actually be quite helpful and help in potentially in deciding who should be in a surveillance program for HCC. Here's a model for alcoholic cirrhosis patients. And this, besides having the patient risk factors, actually has a genetic risk factor. So it has a variant in the PNPLA3 gene that has been included as one of the risk factors. And what this score does is allow us to stratify people as having low risk, intermediate risk, or high risk. And you can see over months, this is about 10 years, substantial differences in risk in this group of patients that are conferred by this risk score. So next, we'll talk a little bit about surveillance in early HCC detection. And the first study I'll do is show you this work that we did a number of years ago as part of the Early Detection Research Network to test the performance of alpha-fetoprotein in combination with DCP, or des-gamma carboxyprothrombin, as well as AFP-L3, which is lectin-binding AFP, or in early hepatocellular carcinoma. Now, it's interesting that we were doing this about 10 years ago or so. We did this study because these 3 biomarkers have actually been used in Japan for over 20 years. And it's actually somewhat paradoxical that DCP was actually first identified at in the intramural program at the NCI. But it's really interesting, and I think it is something to learn about biomarkers, that in Japan, our colleagues there were able to identify DCP as valuable for them in their surveillance protocol. They were able to identify the conditions under which it was most helpful to use DCP. When for a fairly long time, we were trying to use DCP assays that were available to us in the United States and didn't find them to be that helpful. So looking at the performance of AFP for early stage HCC, this is what we found in this collaborative study, that AFP had an area under the curve of about 0.8. And if we think about it at, let's say, 90% specificity, you can see that the sensitivity for diagnosing HCC was just above 50%. if we looked at the other biomarkers in addition to AFP, you can see here that AFP L3 only had a performance 90% specificity at about 40%, and DCP only had a performance of about 35% or so. If we combined AFP with AFP-L3, we didn't really see any substantial improvement in performance. But if we combined AFP with DCP, we did see some improvement in performance, so that 9% specificity gave us about 60% sensitivity for diagnosis of early HCC. So good, but really not ideal. Then in 2014, Philip Johnson, who's an oncologist who worked in Hong Kong for many years and then moved back to the United Kingdom, collaborated with a number of his colleagues in Asia and in the United Kingdom and developed this score that's called the Galat Model. And so Galat stands for gender, age, AFP-L3, AFP, and DCP. And these are the variables here, and you can see, the different I'm blanking on the term coefficients for each of these variables, and you can see the odds ratios for changes in these variables. And so this is the way you calculate the Galat score. And what Philip Johnson and his colleagues found was that the Galat score actually puts you where the AFP, the combined Galat performance was typically above 0.9. So it's around 0.92, 0.93 in most of the cohorts that they studied. And so we decided in a collaborative group here to study the Galat score, and we actually, in addition to looking at Galat by itself, also did a comparison with liver ultrasound and proposed a combined score of Galat with liver ultrasound. So we studied the combined performance of ultrasound and Galat in 111 HCC patients and 180 controls who had cirrhosis or chronic hepatitis B. We found the area under the curve of Galat for HCC detection was 0.95, and it was higher than the area under the curve for ultrasound alone, which was 0.82. For early stage HCC, BCLC 0 and A, which really is when we want to detect liver cancer, Galat was 0.92, ultrasound was 0.82. We found that ascites and a high Child-Pugh score compromised the performance of ultrasound, but not the performance of Galat. And if we added ultrasound and developed a new model, then the combination of Galat plus ultrasound actually achieved an area under the curve of about 0.98. And so this is the performance of the Galat score in all HCCs here with an area under the curve of 0.95, and in early stage HCCs 0.92. And then this is the performance of the Galadus when we added ultrasound. It was 0.98 in all, and in early stage 0.97. So I'll switch a little bit now and tell you about a project that we've been doing with colleagues over the past about 4 or 5 years. And this project is interesting in its genesis because one of our colleagues, Dr. David Alquist, some of you will know him as the one of the main developers of the Cologuard test, which is currently used. It's a stool, it's a combined stool test, which includes stool DNA as well as blood in the stool, and that is currently FDA approved and used for detection of advanced colon polyps and colon cancer. During the development of Cologuard, David was using these two assays to try to detect colon polyps. At one point, the FDA asked him to perform studies to show that his assay was specific to colon polyps and cancer and did not detect other types of cancer. And so David contacted our group and said, is it possible for you to collect stool specimens from your patients with liver and biliary tract cancers that we can assay to show that our assay actually does not detect those cancers. And a few months, and actually probably a year later after we started doing these collections for him, he stopped me in the hallway and he said, "No, Louis, it's extremely interesting. You know, we've been able to select biomarkers, differentially methylated DNA markers that can specifically detect colon polyps and colon cancer and not detect your cancers of interest. But interestingly enough, we were able to also detect differentially methylated markers that can detect your cancers in about half of your patients. So actually, they could detect hepatocellular carcinoma and biliary tract cancer in about half of our patients using stool DNA testing. And that was really the genesis of this project, because then we said, well, if we can detect in stool, then we should hopefully be able to detect in plasma, because plasma, of course, or the liver is exposed to a lot of plasma. You know, about a quarter of the cardiac output goes through the liver at every cycle. And so tumors are really bathed by the cardiac output. So that's where we started. And we started really with the process of discovery, which started with the use of reduced representation by sulfide sequencing. So we started with frozen tissues that we had collected from patients with HCC with cirrhosis, patients with HCC without cirrhosis, and patients with cirrhosis without HCC and controls without cirrhosis. We did the reduced representation by sulfide sequencing on DNA extracted from frozen tissue and identified a total of 302 differentially methylated regions that looked like they were specific for HCC. They went through a technical validation process where we took those same samples, but we did methylation-specific PCR and asked ourselves, can we use methylation-specific PCR assays to identify these differentially methylated regions? And we're able to identify 30 of these regions that we could validate with an assay using methylation-specific PCR. Next, we did a biological validation with a larger number of samples. So we extracted DNA from formalin-fixed paraffin-embedded tissues from a larger number of patients, now about 35 to 40 of HCC with cirrhosis and each without cirrhosis, and about half the number of individuals with cirrhosis without HCC and controls without cirrhosis. Identified that 27 of these differential methylated regions still passed the test, and then went on to a phase 1 plasma study. And now we use a much more sensitive assay, a quantitative allele-specific real-time target and signal amplification assay, or we call it a quartz assay, on DNA again extracted from plasma, using new samples now and looking in patients with cirrhosis and HCC or cirrhosis without HCC. And finally, we did a phase 2 plasma study with the 6 best differentially methylated regions to look at the performance of this assay, and used yet a better assay called TELCUS assay with higher sensitivity for detecting these differentially methylated regions. And I've actually gone through these next couple of slides. These are just showing that slide in more detail. So this shows you the results for the 27 tissue methylated DNA marker candidates. And you can see here, methylation is shown here with the bright red being the most highly methylated and black being unmethylated. And you can see here that the top markers are preferentially highly methylated in either non-cirrhotic or cirrhotic HCCs, but show very little low or no methylation in normal or cirrhotic controls. This is the plasma study now using the quartz and the TELCUS assays. And you can see here that the phase 2 validation for all the full data using the 6 biomarkers. You can see here the performance of the data, the 6 biomarkers together, 0.96, and doing a cross-validation was 0.94. If we looked at the markers by themselves, it was 0.94 here, and AFP's performance was by 0.8. So it was similar to the other study that we had done as part of ETRN. And if we did a combination, we got a little bit better performance of the combination than the methylation markers alone. If we looked at performance by BCLC stage, you can see here that performance did improve by stage, but it was still pretty good in very early and early stages. Particularly early stage performance was quite good. And again, you can see here sensitivity. It was in the order of around 75%. And then going on to, with advanced cancer, it was 100%. So I'm going to switch tacks a little bit here and talk about a recent study that we've done looking at these same biomarkers, but asking the question, looking at, the biomarkers AFP, AFPL3, and DCP, not the differentially methylated markers. So the differentially methylated markers, to close the loop on that study, we've actually advanced to a much larger phase 2. We've done a global phase 2 study, and that work is progressing very nicely and looks as though those biomarkers have performance that is equal to or better than the Galat score. And I think we published some of that work, and some of the rest of that work is currently under review. But I wanted to come back to the idea of genomics now and show you a little bit of data about the genomic associations of the Galat biomarkers. And so this is looking at these biomarkers in a cohort of patients who were part of the TCGA project, which is funded by NCI and the National Human Genome Research Institute. A fair number of those patients in North America, our group at Mayo was actually the largest contributing site to TCGA. We ended up contributing around 65 samples to the project that ended up with a total of about 370 or 380 specimens. And in addition, we had a couple of collaborating groups in Korea that also contributed about 100 of the samples that are in TCGA. And so what this project was we asked which of the patients that had tissue that went into TCGA, did we have samples for, serum samples for in our biobanks? And between our group and the group in Korea, we assayed those serum samples for the AFP, AFPL3, and DCP, and then asked the question, what do we learn about the genomics of these cancers and the associations with changes in biomarkers? So here you can see, the AFPL3, log of AFP, log of AFPL3, and log of AFP. You can see those tend to track fairly closely. Of course, AFPL3 generally is a subset of AFP. So while there are some patients with low AFP that will have high AFPL3, in general, they tend to track together. On the other hand, you can see that AFP and DCP don't really track that closely together. The R was 0.37, and AFPL3 and DCP don't track that closely together either. And in fact, when we look at these biomarkers and we look at patients who had elevations of AFP, in this Venn diagram, AFPL3 or DCP, you can see there's a substantial overlap between AFP or AFPL3 elevation, but not as much overlap with DCP. So the patients, a fair number that have high DCP without having elevation of AFPL3 or AFP. And we of course have a number of individuals too that have normal levels of all three biomarkers. That's why these biomarkers are not perfect, particularly with early stage disease, which is what most of these patients had. And so next thing we did then was we asked, what are the genes that are upregulated in the tumors of the patients that had high levels of AFP, DCP, or AFP-L3? What are the overlaps in the upregulated genes? And what are the overlaps in the downregulated genes? And then next we asked, what's survival in these different groups? And we put them into 4 groups. We had a group of those individuals that had normal biomarkers, so all biomarkers normal. We had ones that had elevation of AFP, L3, and or AFP. When we had that, ones that had elevation of DCP alone, and ones that had elevation of all 3 biomarkers. And if you look at survival of patients who all had resections, presumably for early stage disease, you can see those who had no elevation of any biomarker had exceptional survival. You can see here, we're going out to 10 years, exceptional survival. Those who had elevation of all 3 biomarkers, even after receiving resection with curative intent, had median survival of about 3 years. And those who had elevation of only the AFP or L3 or only DCP were intermediate, still had exceptional survival. They had 5-year survival of over 80%, but you could tell a difference. So what are the genomic associations of these Galat biomarkers? We looked here then at the typical genes that were mutated in these tumors that had these different biomarker profiles. And you can see here, so the common genes, of course, that are mutated in HCC, the 2 most common ones with mutations in the coding sequences are TP53 and beta-catenin CTNN B1. And we go stand from there. And just looking at these first 2, you can see that there are some differences, but there are tumors in all groups that have TP53 mutations, but it looks as though proportionally tumors that had elevations of all 3 biomarkers or of AFP and L3 were proportionally more likely to have TP53 mutations. On the other hand, those who had normal biomarkers for all 3 were more likely to have beta-catenin mutations. And also we had a subset that had high DCP that also had more frequent beta-catenin mutations. And you can look also at the specific profile of the mutations and see some differences as well. These were reflected in pathways, and these are different pathways that were activated or downregulated in these tumors. And you can see, for example, that patients with high AFP, AFP-L3 had particular upregulation of particular pathways. Patients with high DCP had downregulation of particular pathways as well. If we looked also in terms of taking a profile that was derived from the different subgroups. So we derived a profile that was, in a sense, a genomic profile for patients who had RR, who were normal for all 3 biomarkers, for high DCP, high ORL, and so on. And then looked at the entire TCGA data set and said, what are the characteristics of the tumors that are like these tumors? What we found is that you can separate these tumors based on the biomarkers. You see some differences in etiology, for example, and you also see differences in outcome. So that once again, those individuals that were RR-like had the best outcome compared to those who were not. And if we look, for example, at beta-catenin variants, you can see those with RR-like tumors did better than those who had the DCP-like tumors. I'm going to skip this one and summarize here that if we look then at these different characteristics in terms of variants, molecular pathways, and clinical phenotypes, and look at some of the different published subclasses as well, you can see actually distinctions between these different biomarker subtypes and the characteristics of the tumors. And some are good prognosis here, for example. Here, for example, you have a beta-catenin group that's good prognosis, but here, a beta-catenin group that's associated with poor prognosis that is sort of biomarker driven. So I'm going to, I think, go ahead and skip through this in the interest of time. This is just to make the point that In cholangiocarcinomas, we have actually, in some ways, further along than in HCCs. We, for example, identified in intrahepatic cholangiocarcinomas these FGF receptor fusions. This is one of our patients who had FGF receptor fusion with metastasis that we were able to grow as a xenografts in mice. And so you can see these xenografts. This patient had an FGF receptor fusion to the DC6 gene. We were able to confirm this with a break-apart fish assay, and we were able to actually test different drugs and drug combinations against these xenografts. And here you will see, for example, that in the 3 different drugs that we tested, BGJ398 turned out to be the most effective. And this drug actually was just approved, I think, last week for use in intrahepatic cholangiocarcinoma patients with FGF receptor fusions. So there's a potential for genomics and biomarker assays to help us with developing the optimal tests for patients as well. So just to summarize, optimal HD biomarkers for surveillance hopefully are low costs and can be used at point of care, can be used for population, community-based surveillance, and hopefully we can develop predictive biomarkers as well that will help for optimal treatment selection. And I'll stop there and hopefully we have a few minutes for questions. Thanks very much.
Oh, thank you so much, Luis. This is actually very impactful, lots of data, and also speak a very difficult research topic about biomarkers, which probably one of the most important for cancer research, and be able to detect the cancer early. So I guess while we are waiting, probably I have a few minutes for questions, and I would probably going to start one question, which we have actually discussed from previous talk about using imaging as a way of improved diagnosis. So particularly LIRADs. And so what is your thoughts and what's your efforts in terms of using imaging, particularly combined with circulating biomarker for early detection?
Well, it's an excellent question because, of course, imaging is critical once you begin to think about treatment, particularly because it's one thing to know that a person has a cancer. The question is often, where is the cancer and can we treat it? And I think what we continue to pursue are ways to better identify tumors in the liver. There are, I think, some of the challenges we face thinking about imaging from a population perspective are the cost of imaging. So one of the issues that we're trying to address with that is abbreviated MRI sequences, for example, where we try to optimize the sequences that are used so we can image in the shortest possible time. Because then it's a time that equates the cost of an MRI, for example.
I guess the challenge is, I mean, I like your outline is the ideal situation to define biomarker and then also suboptimal, what we are doing now. And I would say majority of the study, or probably if not all of the study, are suboptimal. Most of the biomarkers are just not optimal enough in terms of detecting. Part of reason is that those markers are tumor related and there are a lot of variation, right? How do you see that with the futures? I mean, you outline a little bit, but still tumor marker centric. How do you see that what future would be in terms of really finding some sort of more stable biomarkers, perhaps that will be used for early detection?
I think actually it's very promising because as more work is being done, we're seeing that using some of these other techniques that are developing, looking at different analytes, we're able to improve the performance of some of the established biomarkers we have by adding additional ones and potentially building models that end up giving us higher performance. I think broadly, I think that we're in a good trajectory. I think the challenge is translation. is that, it's sometimes quite difficult to go from a nice biomarker in the laboratory to something that you can actually have FDA approved and use on patients. And that's, I think, part of the challenge.
So we have one technical questions from the audience, and who asks where methylation marks in the promoter region or in the exon site or coding region.
That's a great question. And actually, it was one of the reasons we use the reduced representation by flow of bisulfite sequencing, because it is really agnostic of the location. And so we were able to interrogate, actually, I think the initial interrogation looks at about 30 million different differentially methylated or methylated regions. So it allows you to look both at promoter regions and in exomes or even other regions within the genome also.
Okay, and Eugene has more general questions. You probably read. Can you deconvolute the screen gap into attributable factor for further refined estimate of the screen benefit?
Yeah, so that's a challenging question, Eugene. I think, there's different factors for sure. So for example, if we look at performance or outcomes in Taiwan versus Japan, that's sort of the interesting and obvious one, is that it looks as though long-term people with chronic hepatitis B actually do better than people with chronic hepatitis C. If you look long-term, comparing the Taiwan data to the Japan data. And so there's clearly going to be differences in different regions. And I think there's also differences in different regions because, for example, Galat has age as a cofactor when you use it for diagnosis. And so in the studies that we have initiated in Africa, we already know that we are likely going to have to modify Galat because, the typical age at diagnosis in North America and Europe is in the 60s. And the typical age of diagnosis in Africa is in the 30s and 40s. So if we add age in, there'll be a difference there.
Okay, we have we're gonna just entertain one more questions, and then we're gonna let you leave. So this question from Paul Hayash, who asked that don't you think the surveillance gap you show is depending on the national screen for HBV and HCV in Japan and Taiwan in the first place, which is different from HCC screen and may not translate well to United States and Europe, where NAFLO is the major factor?
Paul, that's a great question. And I think the answer is yes, and so in a sense, doing a national screening, I think we still need to do a national screening program for HBV and HCC to that degree. But NAFLD is a much more difficult challenge, particularly because we see all these patients with NAFLD without cirrhosis who develop HCC. So we know necessarily at the current state that there will be patients with NAFLD without cirrhosis that we would not be able to detect with our current model of screening. However, I think where we are going in terms of biomarker screening is we are going towards screening for multiple cancers at the same time. And I think what we anticipate seeing is that these tests that we're developing for HCC, for example, and the program that we have here includes assays to test not only for things like colorectal cancer, but for lymphoma, for pancreas cancer, for multiple other cancers. So what that does is that it makes it easier to justify putting someone in surveillance because you know that their risk of developing some cancer is much higher than their risk of developing any one cancer. And I think that's the direction we'll likely go.
Very well said. I think it is fantastic. I just want to check with my colleagues, Anu and Tim, if they have any additional thing before I closing.
No additional questions. Just thank you, Louis, for a great presentation today.
Very nice. With this, I'd like to close this session. And thank you so much again, Louis, for coming over to give a very wonderful talk. Thank you.
Thanks very much.
Thank you.